they are able to and grow under different conditions. It's fundamentally tied to concentration of GTP. What happens is a GTP-bound alpha-beta dimer has a conformation that allows it to spontaneously add itself to the end of a microtubule assembly. Now, at some point, that GTP will spontaneously hydrolyze to GDP, not under the influence of a catalyst or an enzyme like GAP, but simply because it is a favorable reaction, and eventually it will happen all by itself. Now, when that happens, as long as there are still GTP-bound subunits at the tip of the microtubule, the remaining portion of the microtubule will remain together, even though they have lost their GTP to GDP. So what we're seeing here on the left is a situation where all of these alpha-beta dimers had GTP when they first added, but they have since had that GTP hydrolyzed to GDP. However, as long as there are still what we call a cap of GTP alpha-beta subunits still adding themselves to the end, these here are able to hold the overall conformation such that the microtubule remains. If, however, we begin to either slow the rate of new GTP addition or stop it altogether, such that the slow movement of GDP-bound subunits towards the tip actually reaches the tip, then we enter a period of disassembly, or what we call shrinking. So the cell can allow or cause these microtubules to grow by making sure there is an abundance of GTP and therefore an abundance of GTP-bound alpha-beta subunits, or it can allow them to shrink by simply reducing or eliminating the availability of GTP, such that the GDP portion will catch up to the tip and it will begin to shrink until and unless the GTP concentration is restored. All right? Now what we're going to see in just a little bit is that once a microtubule reaches a particular length, there are other ways to kind of cap it, to fix it at some particular length, and remove it from that GTP dependency. We're going to look at that here in just a second.
Mm-hmm.
So we have these microtubule-associated proteins that can come in and bind and stabilize these microtubules against disassembly once they've reached some specific length. So we can use the GTP concentration to cause these microtubules to grow or to shrink to achieve a particular dimension, and then we can bring in these accessory proteins to stabilize them. They also can bind along the lengths of these microtubules and kind of mediate attachments with other cell components, whether they be other microtubules in some cases, or the plasma membrane, or the membrane of an organelle. So we have what we call side binding proteins and what we call capping proteins. So we're going to now move to another filament type which has a lot of similarities to microtubules. These are the actin filaments. Actin filaments are made up of actin monomers. These are thinner because they're just kind of a single strand, a little bit of a helical dimension to it, but they're not hollow tubular structures like the microtubules, so they're thinner. Now you might think that that would make them more flexible. They're actually less flexible than microtubules because of the way the individual monomers associate with each other. Like the microtubules, they can be very stable with the addition of proteins that stabilize their lengths and their structures, but they can be very short-lived as well if those particular proteins are not in place. They also have plus and minus ends meant to distinguish them chemically from one end to the other. It's not an electrical identity. Actin filaments, much more than any of the other filaments, can be arranged in all sorts of higher order structures by linkages between other actin filaments at all different geometries relative to each other. So an individual synthesis or synthesis of individual filaments is very similar to the microtubule in that the monomer has to have a particular conformation, in which case it will spontaneously add itself to the end of a growing filament. In this case, though, that conformation is maintained through binding with ATP as opposed to binding with GTP. Just like the nucleation step, the initiation step for microtubule assembly, the first few actin monomers that associate with each other to start the growth of a new actin filament is a relatively slow process. Doesn't involve the same kind of central region that we find with microtubules, but we will see in a second there are some proteins that function as kind of anchoring substrate upon which the actin filament can get started. Once that nucleation step is complete, we do also have this rapid elongation determined by the concentration of ATP-bound actin monomers. And then at some point, the ATP will spontaneously hydrolyze to ADP, just as we saw in the GTP microtubule association. That does lead to the weakening of the filament unless accessory proteins have been added in order to reinforce and stabilize the filament. So the nucleation in this case not happening at the central region, as is the case with the microtubules, but rather at the periphery, at the plasma membrane. So we have a very high concentration of actin filaments at the cell surface. It contributes to what we call the cell cortex. So actin filaments are going to be found kind of directing in two ways. Along the underside of the plasma membrane, kind of parallel to the plasma membrane, to form this kind of mesh-like structure just under the plasma membrane, and then also emanating inwards, kind of going in the opposite direction as what the microtubules are doing, right? Actin filaments are very much involved in the shape, the three-dimensional shape of a cell, and also in the movements of the cells across their substrate, or in a multi-cell organism movement relative to each other in a tissue system. We also see actin filaments participating in the formation of some important structures in animal cells, such as Filopodia, microvilli. We'll see an example of microvilli in a second. Because cell shape is often determined by what's going on outside the cell, cells change their shapes in response to certain types of extracellular stimuli. There are a lot of transmembrane proteins that receive extracellular signals on their extracellular domain, and their intracellular domain is contacting either directly or sometimes indirectly through some intermediaries the actin filaments. And so the result of many of these extracellular signals is a reshaping of the actin filament arrays, which in turn leads to a reshaping of the cell itself. These extracellular signals are often transduced into the actin filament behavior by a family of proteins called actin-related proteins. They seem to function at the cell surface very similar to the gamma-tubulin ring complex. So you end up with an extracellular signal contacting one of these actin-related proteins. They adopt a shape, a conformation that attracts a couple of these ATP-bound actin monomers and facilitate that nucleation step, which can then transition into elongation, and we end up with a new actin filament from that particular source. Now, in the case of actin, we have a little bit more control over this ADP-ATP conversion. We have two different proteins that we refer to as actin monomer binding proteins, meaning they interact with the individual actin monomers. The first of these is thymosin. Thymosin is able to bind to an actin monomer that has not got its ATP bound to it, but instead is a monomer that has had its ATP hydrolyzed to ADP, and it prevents ADP-ATP exchange. So if we were to make an analogy to the kind of GAP-GEF functions involving GTPases, you can think of thymosin as kind of an anti-DEF, although it would be an anti-ADF if you wanted to make it completely accurate. In other words, this prevents the ADP from being removed and the ATP coming in to take its place. The counter to that is a protein called profilin, which is very much analogous to GEF in GTP situations. This promotes the ADP-ATP exchange. So profilin can bind to an ADP-bound actin monomer and catalyze the dissociation of the ADP, allowing an ATP to take its place, thus creating a new conformation on that actin monomer that allows it to add to the end of the growing chain. So we also have a large family of what we call actin filament binding proteins. Now there's a teensy bit of potential confusion if you're not careful with actin filaments. In the case of microtubules, the monomeric, or in the case of microtubules, the dimers from which the microtubules are made have a different name than the actual filament. We have tubulin dimers and microfilament. Here the monomer and filament have the same name, actin monomer, actin filament. So it's important to make sure when we're thinking about what's happening that we are clear as to whether we're referring to an activity involving just the monomer or the filament. Right? So we just talked about thymosin and profilin, which are actin monomer binding proteins. Now we're going to talk about actin filament binding proteins. Right? These can be, like we briefly discussed in the case of microtubules, they can be side binding or they can be capping. But in the case of the actin filament binding proteins, we have a much larger family and a much larger variety of the kinds of proteins that might associate with actin filaments. And because of this diversity, we have a huge variety of higher order structures that we see develop in cells that make use of these actin filaments. So we have what we call bundling proteins. We have gel-forming proteins. So bundling proteins, easiest way to kind of illustrate. So these are proteins that just kind of arrange these actin filaments in parallel arrays. And we have all different kinds of bundling proteins. These are just a couple of what I believe the list would be maybe several dozens, if not several hundreds, of different kinds of proteins that all essentially do the same thing in that they arrange these actin filaments in parallel arrays, but they do it in a variety of different ways. So here we have actually a dimerization where the proteins have an actin binding domain, and then they have a domain that binds to another protein to kind of create a bridge, if you will, between the two. This is a monomeric protein that has two actin binding domains, one on each side. You can see that this protein arranges the actins fairly close together, whereas this one arranges them a little bit further apart. But what's not shown here, but is important to recognize, is so this is a two-dimensional illustration which shows kind of a single layer of actin filaments arranged in parallel. But we often have many layers on top of each other, and the different layers may have different types of bundling proteins, so that one layer might have the actin spaced this far apart, and the next layer might have it spaced further apart. We might actually have one layer where the parallel array is going this way, and the next layer is going this way, or at an angle, all sorts of arrangements in 3 dimensions. And the outcome is a great variety of functions to these. Some of them are structural, some offer some filtration capacities because of the chemistry of the proteins themselves, combined with the actin filaments. Sometimes they end up producing what we call selective sieves, so that some molecular materials can pass through this array, whereas others cannot. Sometimes the trapping is meant as a way to exclude material from the next side of the layer. In other cases, it's actually meant to attract and capture it to feed it into some process. So just all sorts of complexity potentially associated with these different types of arrays. These are what we call gel forming. These are similar to the bundling, except these are able to create a single layer where the actin filaments are not simply parallel, but are arranged relative to each other in varying degrees of angle. Here it looks like it's approximately 90 degrees, but you can see there's even a little twist here. And a very important feature is that there's kind of almost a basket weave element to some of these. So again, lots of different kinds of proteins. These are actin filament associated proteins, and they end up allowing the cell to create all sorts of complex three-dimensional structures using actin filaments as the fiber, if you will, the thread from which these fabrics are constructed. And it's really very much like a fabric, a multi-layered fabric made up of actin filament thread, if you will. So this is an interesting structure that combines a couple of elements. It's shown in 2 dimensions, but this would actually be more of a 3-dimensional structure like a finger. And so you wouldn't have just a single layer of actin filaments, but you'd have multiple layers of actin filaments of parallel rays. So this is a plasma membrane on the outside here. This would have previously been just a relatively flat section of plasma membrane. Extracellular signal in the form of, let's say, glucose, just to make it simple, in the lumen of the gut has contacted a extracellular domain that's converted the signal into the intracellular domain, which has picked up a couple of the nucleation proteins, and we've started growing these actin filaments outward. As they've been growing outward, ER has been very busy delivering vesicles of plasma membrane to accommodate the growth of the plasma membrane that must go along with the growth of these actin filaments. We've added some bundling proteins to connect the parallel arrangement of actin filaments together. We've added these capping proteins here, which are going to be associated with the actin filaments as well as proteins embedded in the plasma membrane to kind of anchor them. And then this region at the very tip, which is going to be made up of a large population of proteins, which not only reinforce the tip, but also cap the end of these actin filaments, which would remove them from their ATP dependence and just fix this length. And the microvilli is kind of a really interesting example because it kind of shows how many different things are all happening at the same time. We're growing actin filaments, we're delivering plasma membrane in the form of vesicles. And all of this is happening simply because you ate a donut. And when that donut is digested, or when the material from that donut has moved through that phase, this entire structure is going to absorb and collapse, presumably because the material is needed elsewhere. And rather than keep the villi there all the time, which you would think would be what you want to do, because you're kind of eating fairly often. You think that they need to be there all the time, but they aren't. They actually go out and come back. You almost think of them like maybe the polyps of a coral or an anemone, where it's just easy enough to send them out when you need them and then pull them back when you don't need them. So anyway, microvilli, a good example of the very complex arrangements that these actin filaments can assume. I'm going to actually flip through this. It's an easy question anyway, which I know is probably good for you guys. So the last one we're going to talk about are intermediate filaments. Intermediate filaments are actually a little bit different than the first two we just talked about, the microtubules and the actin filaments. For one thing, they're not found in all types of animals, whereas microtubules and actin filaments are. Intermediate filaments are only found in vertebrates and soft-bodied organisms such as mollusks. They are structurally very different from either microtubules or actin filaments. They're really arranged into a rope-like structure. They're extremely strong, although rope is probably not completely accurate, because if you take a piece of rope or even a piece of wire and you bend it and let go, it kind of stays the way you left it. Intermediate filaments are flexible like that. You can twist them, you can turn them, you can tie them in knots, but when you let go, they return back to their original dimension. So kind of a lateral spring, I guess, if you will. Their function is to provide mechanical strength, right? So the reason we see them in soft-bodied organisms is relatively obvious. They do give these organisms free of any skeleton structure the ability to maintain some shape and some integrity. Insects, for example, with an ectoskeleton do not need these types of structural supports, and so you don't see them in insects. Large animals, however, with a skeleton such as vertebrates, it turns out that our soft tissues are far enough away from our internal skeleton that we do need that mechanical support. So kind of think of that space between your skeletal system and the periphery as being not much different than the body of a mollusk or a soft-bodied organism, which is why we find them in vertebrates. So, unlike actin filaments and microtubules, which are constructed from globular monomeric proteins, intermediate filaments are constructed from long helical proteins that have this very long helical domain, all right? Then two of those are wrapped around each other to produce what's called a coil. When we say alpha helical region, what are we referring to when we say a alpha helical region? What would this region of this protein be comprised of?
GDP?
No. What gives it its alpha helical structure? Why are they calling this an alpha helical region?
The twist.
Hydrogen bonding between what?
The two separate strands.
No, this is just a single strand. This is a single protein. Primary structure. This is just nothing but a string of amino acids, but they're calling this an alpha helical region. What is the alpha helix that they're referring to? We talked about it way back when we talked about basic protein structure. Hydrogen bonding is what maintains that alpha helical structure.
It's amino acids.
Well, it's amino acids, but what in particular? What is exactly, what is
The
this?
hydrogen bond holds the parallel and the anti-parallel strand. That's the DNA.
There is no parallel, anti-parallel
There's not.
strand. This is a single string of amino acids. Not the side chains. What are the four structural levels of a protein? What's the first level of organization? The first structural level is referred to as the what?
Primary.
The primary. And what's the second?
Secondary.
Secondary. And what is secondary structure? What are the two different types of secondary structure? Alpha helix and
Beta sheet.
beta sheet. And what holds secondary structure in that structure?
Hydrogen bond.
Between hydrogen bonding between what?
Based between
Side chains.
What?
Side chains.
No, not between side chains.
The amino and carboxyl group.
What's that?
The amino and carboxyl group.
Of what?
The amino acid.
But what is it that where the amino and the carboxyl come together? What do you call that?
Hydrogen bond.
No,
No, that's okay.
that's the peptide bond.
Sorry.
It's hydrogen bonding between the electronegative oxygen of one peptide bond and the delta plus hydrogen of the next peptide bond. Every pair of peptide bonds, if the geometry as defined by the amino acid side chains allows for it, are attracted to each other. And when they are attracted to each other, if they are, sometimes they aren't, they're not able to do this because the side chains don't allow it. But in those types of structures where it's allowed, they arrange themselves into either alpha helices or beta sheets. And that's our secondary structure. It's extremely strong and extremely important in certain circumstances, and this is one of those circumstances. So every single pair of peptide bonds along this entire length are hydrogen bonded to each other. And we take two of those and we wrap them around each other to form this coiled coil. And then we arrange a bunch of them end to end to produce these very long kind of superfilaments. And then we take 8 of these superfilaments and we twist them around each other just like a rope or a cable. And the result is an extremely strong structure. Because if you think about the actin filament or the microtubules, the interface between monomers is just this little tiny surface where the globular structures come together. So you might have a couple of dozen weak non-covalent interactions. Here, you've got this entire length of surface held together with weak non-covalent. So if we just put a number on the one on actin or microtubule, let's just say there's 50 weak non-covalent. If there are 50 weak non-covalent, in that case, There are thousands and thousands here. It is just a substantially stronger structure, even though it's based still on the same individually weak non-covalent association. There are just so many more of them. And when you put it together like this, you end up with this really super strong structure. All right, so vimentins provide mechanical support in glial cells. Remember we talked about the myelin sheath that surrounds nerve axons. That's a very long, thin cell. It is supported by a type of cytoskeletal element called a vimentin. So which category, which class of cytoskeletal element would you think that is? Intermediate filament, microtubule, actin filament, or myosin, or myelin?
Yes.
All right, so a type of cytoskeletal element that provides mechanical support would be what type of cytoskeletal element?
An intermediate filament.
Mechanical support.
A.
Intermediate filament. That's the role of intermediate filaments, provide mechanical support, right? So there are going to be lots of individual names for these different filaments. So vimentins are a type of intermediate filament. All right, so we have three different types of cytoskeletal elements: actin filaments and microtubules, which are common to all animal cells, all cells that lack cell walls. Intermediate filaments, which are found only in a subset of those types of cells, those that are either soft body or vertebrates, where there is a large region of soft tissue that extends beyond the support of the skeletal system. Then associated with these filaments, we have a variety of motor proteins. We do not have any motor proteins that associate with intermediate filaments. They only associate with either microtubules or actin filaments. They're powered by ATP hydrolysis. Despite the variety and different evolutionary histories of the different motors, they all use repeated cycles of ATP binding and hydrolysis to force conformational changes that are energetically favorable, so energetically favorable that they, if they are attached tightly enough to other structures, are able to drag things along with them as they undergo these conformational changes. It allows them to typically step along a cytoskeletal filament, but we have a couple of different types of actions. We can have what we literally think of as a walking along a cytoskeletal filament, dragging something along with you like a vesicle, but we also have situations where they can be attached to the plasma membrane and they walk along the filament, essentially pulling, stretching, reshaping the plasma membrane as they move. We also have situations where two motor proteins will be attached to two different elements and attached to each other and can work together. If one's trying to walk to the left and the other's trying to walk to the right, they can't do that. They stay stationary, and instead the filaments move under them, and they can move these filaments in opposite directions. So they have a head region, which is also what we call the motor domain. This is a relatively small, globular, compact region. It is the region that binds and hydrolyzes the ATP, but it also has the ability to attach itself to something, either a cargo like a vesicle or a plasma membrane, or maybe the head region of another motor protein. And then we have the tail region, which again is the region that's going to attach itself to something else, again, a cargo possibly, a plasma membrane, or even another motor protein. The tail region is typically long and flexible, and what ends up doing is when the head region undergoes a relatively small conformational change as a result of ATP hydrolysis, this very long tail region, because of its kind of lever activity can actually cover a great distance. And so you get this amplification of the amount of movement where the cargo is attached relative to the tiny movement that occurs really at the point of the actual conformational change. So there are lots of different kinds of actin motors. The most common are what we call the myosins. And there are many, many, many kinds of myosin motor. There are only a couple that we are probably familiar with. One or two in particular that are very well described and are kind of used as the poster child, if you will, for myosin motors. But there are hundreds and hundreds of different kinds of myosin motors. Myosin 2 is the one that is associated with muscle contraction, and this one is the one that is most well understood and is often used as an example of motor protein function. There are so many myosins, in fact, that the details of how a lot of them operate and the specific circumstances under which they may be utilized is not entirely known. What is known is that they all hydrolyze ATP, and they mostly walk towards the plus end of an actin filament, all right, which is the growing end, if you will, of the actin filament. These are often arranged to allow two filaments to slide past each other. So that would mean that the head regions are attached to two different actin filaments, and the tails of the two motors are attached to each other. We also have microtubule-associated motors. We have two different types, two different families of microtubule motors. We have the kinesins, most of which walk towards the plus end of microtubules. These are structurally very similar to myosin 2, the actin-associated motor proteins. They are presumably evolutionarily related as well. These also function in the formation of the mitotic spindle, and the segregation of chromosomes depends very heavily on the activity of the kinesins associated with microtubules during that phase of the cell cycle. Then we have the dyneins, which are kind of the odd man out, if you will. Dyneins walk towards the minus end of microtubules. Dyneins are quite different structurally than kinesins or myosins, although they still do utilize ATP like all motor proteins do. They're much larger than the kinesins. And they're presumably not evolutionarily related, are in fact an example of convergent evolution. They function in organelle transport, but they also function in the movement of flagella and cilia in eukaryotic organisms. Flagella and cilia in prokaryotic organisms operate very, very differently, have nothing to do with cytoskeletal elements. These are very fast as well, probably due to their larger size. They're able to bind and hydrolyze ATP at a much greater rate. So they're fastest, they're the largest, they're kind of the strongest, if you will, as well. So we're going to look at myosin 2, which is often the motor protein that we look at in order to get an idea of the sequence of events that leads to this energetically favorable movement and the ability for this protein to do mechanical work, to convert energy in the ATP molecule to use or the ability to perform mechanical work. So the default conformation for a so I guess maybe we'll look first and see what we have here. So this is muscle cells. So there's a few kind of interesting things that maybe are a little bit confusing if you first look at it. So this is a single myosin motor protein. This is the head region. So this is the region that attaches to the actin filament. It's also the region where the ATP will bind. It's also the region that catalyzes the hydrolysis of ATP. This is the tail region, or what we would call the cargo region. Now, what's shown here, which is not necessary really for what we're looking at right now, is a whole bunch of other myosin tails. That's what this green is meant to depict. So really, what if this were drawn a little bit more accurately, is there would be hundreds of these myosin 2s, each one attached to the actin just a few nanometers away from the other, and all their tails would kind of all be going in the same direction, kind of coiled together. So we're gonna look at that in just a second. For right now, just looking at the individual myosin motor protein. So the default conformation for myosin and the default conformation for most of these motor proteins is to actually be attached to the actin filament, right? That's why we call this the rigor conformation. It's the reason rigor mortis exists, because in the absence of active ATP binding or hydrolysis, these myosins are all attached to actin filament. If we introduce an ATP molecule, the ATP binds to the head region, it changes its shape, and it causes it to dissociate from the actin filament. But with that dissociation and that new conformation also comes a conformation that activates the myosin's ATP hydrolysis function, and the ATP is hydrolyzed to ADP. That does two things at the same time. The first thing it does is cause a change in the shape of the protein so that the head region is reoriented relative to the tail region, a region maybe a little further up, if you will, than it previously was. It also creates a conformation that allows for a weak binding to occur between the ADP bound head and the actin filament. But notice it's now bound to a region that is a little bit upstream of where it was originally bound. That leads to another conformational change. This conformational change causes the ADP to dissociate, at which point we're back to what we had here. So what happens when the ADP dissociates, the myosin head and tail reacquire the arrangement, the orientation they had relative to each other originally, and the attachment between the myosin head and the actin tightens up considerably, and we return back to where we were. However, because we are now bound here instead of here, the result is that the entire filament moves to the right. So the actin in this case is stationary, and the myosin too, which starts out here, ATP causes it to separate, hydrolysis, rebinding, loss of ADP. And it just keeps happening over and over again as long as you continue to introduce the ATP. Kinesins and myosins, both of which associate with cytoskeletal elements, have two different kind of organizational features that allow them or cause them to be associated in different ways. Kinesins and dyneins have two head domains, and the way they are organized is that there is no point in the cycle of ATP binding, ATP hydrolysis, ADP dissociation, and rebinding. Even though there are points in time where one of the two head regions is not associated, there's no time when both are dissociated, which means that a single kinesin or a single dynein can walk on a microtubule carrying a cargo. It can operate all by itself because it never loses its grip on the filament. The myosins, on the other hand, kind of hop. And because they lose their connection with the filament every cycle, if there were a force opposing their forward movement, as is often the case, imagine that they're trying to stretch a spring from one end to the other. They could stretch the spring, but every time they hop up to get their next attachment point, they would just get pulled back to where they were originally. For that reason, you don't see myosins operating as individual motor proteins like you do the kinesins and the dyneins. They only operate as part of a large group of myosins all working in the same direction, so that at any point in time, a large percentage of the group is attached to the filament, while the other percentage of the group is in its hop stage of the cycle, if you will. So collectively, they're never completely dissociated as a group. So which of the following would most likely involve a minus-directed motor protein? Endocytosis, exocytosis, COP2, muscle contraction, or none of these? All right, so this is not necessarily a super straightforward question, but it's answerable. So we can start just with muscle contraction. Muscle contraction involves what type of motor protein? Myosins. And myosins move towards which end of the actin filament?
Positive.
So would that involve a minus-directed protein?
No.
No. So now we think about endocytosis, exocytosis, and COP2. So we could look at it kind of a clever way that allows you to answer without knowing the answer. You can do something slightly clever. So COP2 direction, COP2 vesicle traffic goes in which direction?
COP2, COP2 goes back to the endoplasmic reticulum.
Yes or no?
I say yes, and the OG2 back to.
Yes, I'm asking other people. Is that right or is that not right? Which direction is COP2 vesicle traffic? Is it going out or is it the retrieval pathway? It's going out. COP2 goes out. COP1 is retrieval. So COP2 is going out. What other here is going out?
Exocytosis.
Exo. So B and C are really the same thing. They can't both be right. So we're actually looking at endo, right? Now, where is microtubule anchored? Where do microtubules originate?
Inside the cell.
Oh, we got a lot of studies. Where do microtubules originate? From where do microtubules originate? Where do they get their start?
The centrioles.
What's that?
The centrioles.
Centrioles. Right? Remember the sea urchin? That's what the astral conformation kind of looks like. They start at the centrioles and they grow out from there. So which end of the centrioles, which end of the microtubule is anchored in the center? The minus end or the plus end of the microtubules? Well, what direction do they grow? Do they grow out in the minus direction or do they grow out in the plus direction? I want someone to say it with conviction.
They're connected in the minus end, they go out in the plus.
They grow out in the plus direction. They're anchored in the minus direction. They grow. The plus end is the fast growing end. They're anchored in the centrioles. They grow out in the plus direction. So endocytosis is a process of bringing things in, which would require a motor protein that is moving towards the minus end, which is towards the center of the cell. So endocytosis is a process that involves a minus-directed motor protein. These three all involve plus. Myosins here, right? Kinesins here. I could ask another question. What type of motor protein is involved in endocytosis? Dyneins. Dyneins move towards the minus end of microtubules. Kinesins are involved in exocytosis and COP2 vesicle traffic. Dyneins are involved in endocytosis and COP1 vesicle traffic. Myosins are involved in muscle contraction, as well as a bunch of other things. So there's like 10 possible test questions there. So yeah, a lot of you got it right, but you couldn't explain why you picked that one. maybe just shy. So when we look at muscle contraction as kind of an example of the really elaborate processes that this muscle, motor proteins can mediate. So muscle cells are kind of interesting cells. A muscle cell is a fusion of hundreds of individual cells. So through embryogenesis, differentiation, hundreds and hundreds of individual cells eventually become fused together into these very long cylindrical cells. They retain their nuclei, but most of the rest of the cell is transformed into this very tightly packed structure called myofibrils. So every one of these is a myofibril. This shows that maybe a dozen or so. There are actually thousands of myofibrils, not just however many are shown here. This is obviously an actual micrograph, so this is how many, every one of these little dots is a myofibril. Each myofibril is in turn composed of a long series of individual units that we call sarcomeres. That's what's depicted here by this kind of little disc-like structure. It's a little bit misleading here because this makes it look like the diameter of the myofibril is much greater than the length of the individual sarcomere, but that's really not true. The sarcomeres are actually longer than the diameter of a myofibril. So we have these individual myofibrils made up of a whole bunch of these individual sarcomere structures. Each sarcomere is a parallel array of myosin filaments. The myosin filament here is the long tail region of the myosin motor protein, and actin filaments, which are referred to as the thick filaments. And these sarcomeres represent the individual contractile unit of a muscle cell. It's a very interesting structure. So we have a whole bunch of stuff going on here. We have, first of all, the actin filament itself, which is the little white dots are the actin monomers all put together to form this actin filament. So this actin filament actually was anchored here. It was anchored here originally and has grown out. And so this is the plus side here, this is the minus side here. This Z disc is made up of a whole bunch of proteins that not only form kind of the barrier or the boundary between two adjacent sarcomeres. So this is one sarcomere, this is another one here, and there'll be hundreds and hundreds of them arranged end to end. But it also has the proteins necessary to stabilize the actin in a finished length, all right? We also have capping proteins here, which again stabilize this end of the protein, right? We also have a protein called nebulin, a long filamentous protein that wraps around the actin and gives it a little bit more structural integrity. And then we have type, which is an interesting protein. It's kind of a spring-like protein that attaches to the Z disc at one end, and it's intertwined in all of these at the other end. So every one of these little golf club looking things is a myosin 2 motor protein head. The tail of each of these motor protein heads extends into this region here, and the tails of these extend into that region. So the tails of these two motor proteins are twined around each other. In other words, all of these myosin tails are attached to the tails of these, right? Which means if these want to go to the right and these want to go to the left, they can't, because they're attached to each other, right? Like tying a rope between two cars and trying to have the two cars drive away. Can't do it. But what we have is this kind of this actin filament, right? And what happens is when we introduce the ATP, these myosins grab onto the actin and they try to move towards the plus end, which is to the right. And these myosins grab on to the actin and try to move to the left. But because they can't, what ends up happening is this whole array here and this whole array here are drawn together, right? The two Z disks simply get closer together. And because we've got so many myosins doing that at the same time, there's quite a bit of force generated. And so these two Z disks can be drawn together despite the fact that there could be considerable resistance to that movement. Now you amplify that, and you imagine that you've got thousands of these sarcomeres arranged end to end. And if every one of them shortens by 1 nanometer at exactly the same time, then over the entire length of that cell, you can have something that shortens by 1,000 nanometers. And if you've got thousands of those cells arranged end to end, then everything contracts at the same time, you can shorten by 1,000,000 nanometers, right? And you can actually convert an individually tiny movement into this very large scale movement. Additionally, because you've got thousands and thousands of these myofibrils in each cell, and thousands and thousands, actually hundreds of thousands of cells in each muscle fiber, the amount of strength generated, the amount of force generated is quite substantial, and allows the muscle to actually work efficiently against a considerable load. It's nothing more than the addition of millions of individually short and relatively small force events happening all at exactly the same time. Actin filaments in sarcomeres are very, very stable. They're probably the most stable example of actin filaments that we have. So the way it kind of works in a large, back up and take a look now at a bigger picture even than this, kind of combining a few different things that we've talked about. So how do we actually get from, actually there's one still huge mystery in all of this, which is when you decide to move your finger, why does your finger move? The short answer is we really don't know. We know an awful lot about what happens chemically once the stimulus is initiated, but we really still don't exactly know what initiates that initial initiation, if that makes sense. We do know that it starts with a stimulus from nerve centers, nerve tissue, that generates an action potential. The action potential, which is this flowing wave of high sodium concentration, travels across a couple of synapses with an opportunity for other signals to say, no, don't move your finger right now. And eventually